Hello friends, welcome back to my YouTube channel Nasrin Padasalai. In today's video, we are going to discuss the answer for those questions that have been asked in the subtopic biochemistry. If you have selected this biochemistry, then please watch the full video. I have given you many different explanations regarding the questions that have been asked. I hope few questions will be confusing for you. Let's discuss in the video. If you didn't attend the biochemistry, let me give a try for watching this video and it may help for your further exams like your DBT and your CSIR exams. So, first question is your anti-B antibodies are present in the serum of. It is an MSQ type of question. You should know blood group AB will have both the antigen A and antigen B on the red blood cell, but the blood group doesn't have no antibody. Blood group A will have anti-B antibody in the blood and antigen A on the RBC, and blood group O which will don't have antigen on the red blood cell, but it have both anti-B and anti-A antibody. So the correct option is option number A and option number B. Next thing is that in the plot given below, the solid line represents oxygen binding to hemoglobin under a physiological condition. Broken line represents the condition of. So you should know it is an oxygen binding curve and there is a right shift and there is a left shift. I hope you know about all those kind of things. Maybe I shouldn't give all those explanations to you, but let me give you a few explanation which will be very much helpful for you. Okay, so you can keep this references for your CSIR also. So this is a kind of thing where okay. So, okay, left shift mean it is an increased affinity to oxygen. Right shift mean reduced oxygen affinity. Decreased temperature, decreased in 2,3 bisphosphate glyceride, decrease in hydrogen ion component concentration and increased in pH will lead to left shift. So, what they had asked which has given to left shift. So, high pH will lead to this left shift. Okay. So, the next thing is that a value of a guanidine group of arginine is. So they are specifically asking this guanidine group. So the pKa is 12.50. Which of the following process are energy requiring process? It is your Na plus K plus pump where for each 3 Na plus ion there is 2 K plus ion being pumped. An active transport is also energy requiring process where in facilitation diffusion which is mediated by your channel protein is not the correct energy depending process, and non-minute transport means it is a simple diffusion and it also doesn't require energy. According to Obel hypothesis, inosine at third position of anticodon cannot form hydrogen bond with. Inosine I has a capacity to form bond with cytosine, adenine, uracil. It cannot form bond with this guanine. Okay, so the correct option is option number A. Okay, the non-coenzyme vitamin is your folic acid, when nicotinic acid, your niacin, NAD, ascorbic acid, vitamin C, and for pyrolyl hydrolysis activity which is formed in collagen formation, thiamine pyrophosphate. Next thing, which one of the following molecule do not absorb at 280 nanometer in an aqueous solution of a pH 7 at room temperature? Option is phenylalanine, because phenylalanine absorb around 240, whereas your ATP and polydeoxyguanine nothing but they are the nucleotide related content, so they will observe at this particular nanometer, and tyrosine also having a this particular observance. Okay, and they are responsible for extrinsic and intrinsic fluorescence in the UV spectrophotometer. Consider the following open chain form. Which of the following pair represents a epimer? Already I placed a detailed video regarding this epimer and in many other contents, and the answer are 2 and 4, that is D-glucose and D-mannose, which differ the stereochemistry of carbon number 2. Okay, you all know epimer means it will have a same structure, only at one particular stereochemistry they will differ. Next thing, D-galactose and D-glucose differ in the stereochemistry 4. Next thing is that this is the easiest question with respect to this STS page. Which of the following statement is or true? protein migrate toward anode. Yes, because they are saying STS page. They will impart the negative charge to the protein molecule. So NAR will migrate toward the anode. ETBR is used for tracking. It is not the correct answer. It is a wrong answer. Lower molecular weight protein migrates lower than larger molecular weight. It is a wrong answer because larger molecular weight migrates lower than lower molecular weight. Beta mercaptanol is used to reduce disulfide bond. Yet it is the correct answer. So option number 1 and option number 4. A molecule that form a donor acceptor energy transfer pair with dancel group are nothing but this is aliphatic, that is glycine, valine, isoleucine, leucine, proline or aliphatic, and aromatic is your tryptophan, tyrosine and your phenylalanine. So here tryptophan is the correct answer. Which one of the following antibiotic can form an ionic channel with a bacterial membrane? It is your gramicidin A. It is isolated from a bacterium called Bacillus brevis, and this gramicidin is also have a same function as that of your nalidic acid, and it is example it will allow include this cations, and it will lead to ionophoric media in form force in a gram positive bacteria, and this gramicidin antibiotic is also have a hemolytic activity. For that purposes it is not been given is as an intake. It is generally employed as a tropical agent for many kind of ophthalmologic related disorder. Next thing is that which of the following lipid is non-ionic? It is galactocerebrosid. High energy compound of your 1,3 bisphosphoglycerate and phosphoenol pyruvate, whereas your AMP and vitamin K are not the high energy component. Next thing is that telomerase has a function similar to reverse transcriptase, because your reverse transcriptase also will lead to the form cDNA from RNA. Telomerase also have a same function. Okay, you all know telomerase is a terminal transferase and it will end replicate the DNA. Okay, so your DNA polymerase cannot replicate your entire DNA. Okay, next tertiary phase used in gel filtration chromatography is composed of your spherulos. Other example are frds, everything. The carboxymethylase and diethylaminoethyl are used in ion exchange chromatography. Okay, and also this blue dextrin is used for a wired volume in gel filtration chromatography. So that's it, friends, and I hope I had provided you a detailed related information with respect to this kind of biochemistry. If you like the video, like the like button. Thanks for watching my video, friends. Subscribe, and definitely I will come up with a useful content for you in my next video.